Dear students, in this particular lecture, you will be learning on plant regeneration pathways or routes. So, how this plant will regenerate or renew or it will produce new plantlets. We will see. The basic definition for this is the plant will renew physiologically, and also whatever the damaged parts are there, it will be naturally repaired, and also whatever the damaged plants will be replaced by the new plant by the plant itself. The plant will regenerate and it will renew and it will repair whatever the damaged plant parts. So this can be seen in different ways, like through the callus formation and then root and shoot formation, that is called as direct, and indirect without formation of the callus. So then we'll see one term, that is morphogenesis. It is a biological process in which the cells, tissues, or other organisms will develop different shape or structure. So organogenesis is related to the development of the organ, whereas morphogenesis it is related to the development of plant, like whether the maybe increase in the girth or height or entire plant architecture, that is called as morphogenesis. So under tissue laboratory condition, this plant regeneration pathway can be seen in two types, that is organogenesis and somatic embryogenesis. Organogenesis means here the newly de novo origin of the organs. It may be root or shoot in tissue, like in a cultured tissues. It will start producing the new organs. It may be root or shoot. Somatic embryogenesis, here it will produce the roots, shoots from the embryos, like whatever the somatic cells are there. So from that as well as from the embryos, it will produce the like different organs. And here there are two main growth regulators which play a major role in formation of root and shoot. Suppose if the auxin to cytokinin ratio is high, that will leads to the formation of the roots. Suppose if auxin to cytokinin ratio is less, that will helps in formation of the shoot. And whatever the callus formation, that process is called as callogenesis, and root formation is called as rhizogenesis. These are the two terms which is very important. Organogenesis, I already told, the formation of different organs. So mainly this organogenesis can be divided into two types, direct and also indirect. So direct organogenesis, here the organs will directly form, like without callus formation, directly from the explant to directly it will produce root or shoot. And here the regeneration will be more regeneration we can expect, but the and also same way it will also produces less somoclonal variation. Then we'll see like embryogenesis or embryo. It is a like seed embryo which is a part of the plant. So it will also helps in production of the root and shoot. Then in indirect organogenesis, here the explant first develop the callus. Then after callus formation, the organs will start to produce like root and shoot. Here regeneration we can do from the different part explants, but here the genetic variation will be more, or somoclonal variation will be more. Somatic embryogenesis, here also like same way, uh, but in case of organogenesis, it will produce the monopolar embryos like shoot or root. But in case of somatic embryogenesis, so it will produce complete bipolar embryo or root, shoot, apex. So that is the one main difference. Here normally it works on the principle of totipotency. Totipotency And here also two types embryogenesis we can divide into two types, direct and indirect. Like some from the direct somatic cells it will produce the like entire plant. We can produce genetically true to type plants which are free from pests and diseases. Here, what are the advantages of this direct somatic embryogenesis is callus will be like there will be no callus, we can reduce the time. But here the uh, process can be, we can faster the process, we can avoid the callus formation stage and reduce the somoclonal variation as well. Disadvantages here we can see the rare occurrence will be there, not much successful, and also sometimes it is challenging also. Survival rate will be also very less, and genetic potential will be low. Like potential, we cannot perform in all the crops, and also uh, the multiplication rate will be less. Indirect somatic embryogenesis here, the embryos will first produce the callus, then it will produce the root and shoot. Normally, this can be used for large scale production, and here 2,4-D we can use as a synthetic axin for enhanced production of the like roots as well as shoots. We can use commonly. The advantages are like we can expect more more plantlets, and superior clones also we can use, and we can apply in different kinds of species. Disadvantage here is somoclonal variation. Again, here also it is a problem, and the steps are more here. There are more number of steps, and also sometimes the somatic embryos will not germinate properly. This like physiological immaturity that causes the biochemical immaturity also, and that leads to the ultimate reduction in the germination process. So here you can see the embryo-like structure. Embryoid will form, like embryo-like structure will form, and it will produce the bipolar thing, like bipolar, and there is no vascular connection as well, and we can see the group of cells. And this embryogenesis was clearly explained using cell isolation theory by Stewart and his co-workers during 1964. It refers to the cell separation or cell sorting. This process, it helps in isolation of the cells from the heterogeneous group of a sample. Like whatever the targeted cells are there, that particular cells can be identified here, and also it can be separated like using some physical and physiological parameters. And even the whatever the we can also connect with the plasmodesmata. And another one theory is differentiation theory. Here the explant have to undergo de-differentiation to form the callus. Then again it will the callus will produce the embryos. So like this